I'm excited to present my work on preserving joint function after injury. My research career started at MIT, where I first fell in love with musculoskeletal research in the labs of Al Grodzinski and Molly Stevens at Imperial College. This love took me to the University of Pennsylvania, where my thesis work focused on patterning soluble factors in cells for cartilage and bone tissue engineering in both in vitro and in vivo animal models. Most recently, I've moved to the Biofrontiers Institute, where I've been focusing on developing vascularized disease models of inflammatory joint disease. I hope from my research background, you can appreciate how I have this love of orthopedic research. And for most of my studies, I focused on the knee joint, which is a complex organ made up of many different tissues. I like to say that the knee is similar to a balloon, which is an enclosed space. And so any injury to one tissue often spreads. And this is why so many people worldwide have osteoarthritis, which is a degenerative joint disease. And so I'm really interested in this overarching question of what can we do to preserve joint function after an injury, such as an ACL tear or a meniscus tear. So what do surgeons currently do for clinical cartilage repair? Well, it's actually very similar to repairing a pothole, where the damaged asphalt or cartilage is removed, the hole is patched, and then there's some time that's waiting before applying weight. And so this damaged tissue is biologically and mechanically inferior to the surrounding environment. And so for my PhD, I looked at native cartilage and was really enamored by this structure and thought that if I could replicate the cellularity and mineralization of this tissue at its interface with bone, maybe this structure would impart better function and longevity for patients. And so I developed one strategy to pattern cells to replicate this cell gradient using magnetic fields. And to do this, I injected a magneto-responsive fluid within traditional cross-linkable hydrogels. And here you can see how we're increasing the magnetic field exposure over time, and these unlabeled cells are moving upward away from this underlying permanent magnet. And using this very simple approach, we can not only create cell gradients, but we could create ECM gradients, and also these cells remain viable over long-term culture. To create patterns of soluble cues to create this mineralized interface, I encapsulated mineralization promoting therapeutics within these drug delivery vesicles or double emulsions, which had these very thick shells. And these thick shells enabled these capsules to rapidly settle when injected into saline, which enabled us to deploy these within a preclinical animal model. I injected them within these cartilage defects. They settled along the osteochondral interface, and then cartilage could nicely form on top of the capsules. We found that these capsules improved mineralization at this juncture between cartilage and bone. Moving into my postdoctoral work, I'm still interested in this big picture question, but now looking to improve our mechanistic understanding of disease. Here, I'm focusing on a new musculoskeletal tissue I haven't told you about yet, the synovium, which is a vascularized joint lining tissue. This tissue, interestingly, it has an increase in vascularity and also in cellularity with disease state. And so I've been developing this vascularized in vitro platform that we can use to study how specific cells within the synovium contribute to disease pathogenesis. And so far, we have discovered that the pro-inflammatory cytokine IL-1 beta decreases ECM remodeling around vessels, and this may contribute to the leakiness found in these vessels and also immune cell infiltration within this tissue. Moving forward to my future lab, I'm interested in further developing these disease model platforms to uncover cellular mechanisms of inflammation, develop new injectable therapeutics to treat and monitor inflammation, and also develop new advanced biofabrication techniques to pattern musculoskeletal tissues. With that, I'd like to thank you for listening, and I'd be happy to take any questions.